The overall goal of the following experiment is to study the effects of staphylococcal phenol-soluble modulins, or PSMs, on human neutrophils. This is achieved by first isolating neutrophils from human blood for subsequent in vitro analysis. Next, the neutrophils are either loaded with fluorescent dye and mixed with a concentration series of PSMs with and without inhibitors, or they're allowed to phagocytose S. aureus bacteria containing a PSM alpha promoter GFP reporter construct. Ultimately, activation of the neutrophils by PSM stimulation can be monitored by flow cytometry, and expression of GFP by the phagocytosed bacteria can be analyzed by fluorescence microscopy.
By this method you can study the effects of staphylococcal phenol-soluble modulins on neutrophils. Of course, this method can be applied to many other cell types, stimuli and antagonists.
We first had the idea for the microscopy part of this method when discussing a way to observe the effects of PSMs inside neutrophils. Using a bacterial GFP expression construct and time-lapse microscopy allows us to do exactly that.
After drawing 5 tubes of heparinized venous blood, pour 12 milliliters of a 1.119 grams per milliliter density Phycol solution into a 50 milliliter tube and then carefully overlay 10 milliliters of a 1.077 grams per milliliter density Phycol solution. Next, dilute the blood with an equal volume of PBS and layer 20